Yes. So yes, today we are going to build up the concept cell cycle and cell division. From this chapter, what we have to actually exercise is what are the phases of cell cycle, okay, and what will be the time period of the cell cycle in different organisms. What are the phases in interphase? What are the? We are going to build up the concept cell cycle and cell division. So yes, in the interphase we have G1, S and G2 phase. M phase we have karyokinesis and cytokinesis, that will be different in both meiosis and mitosis. Let us get started. Before getting started, we have to know what sort of questionnaire you will get mostly from this chapter, fine. Rarely or mostly, first we will go with most proximity of questions. Definitely or sure shortly you will get a question from the substages of prophase 1. Okay, from substages of prophase 1 in meiosis. And second type of question is match the following question. Okay. Third is statement or assertion based on the phases and activity done in the each phase. And fourth type you can expect is a diagram based question. Fine. These are different levels of question. And last but not the least, you can expect numerical based questions beta. Numerical based like what will be the number of mitotic divisions, what will be the number of meiotic divisions, what will be the number of meiotic divisions for formation of so and so number of seeds, fruits, grains. This type the questions will be. And most importantly about chromosome number. What will be the chromosome number in G1, S, G2 phases. Like if you have seen the recent EAP set paper, in that the question is asked about chromosome number. Okay. So fine, we will discuss all the concepts and exercise the objective also, including NEET PYQs. So first coming to cell cycle is nothing but the sequential events which has two phases. What are they, Nana? Interphase and M phase. Okay, you have interphase and M phase. You have to know what are the synonyms of interphase. What are the synonyms of interphase? Interphase is also called as preparatory phase, biosynthetic phase or resting phase. What are the three synonyms, ma? Interphase is also called as preparatory phase, or resting phase or synthetic phase. Good. Synthetic phase. Do remember these synonyms. Okay. What are the three subsequent phases in the interphase? G1 phase followed by S phase followed by G2 phase. Okay. G1 phase followed by S phase followed by G2 phase. What are the synonyms for G1 phase? Okay. G1 phase, if you can clearly see, it is after M phase, right? So this is also called as post mitotic phase or pre-synthesis phase. Like that, you tell me what will be the synonyms for S phase. It can be post G1 phase or pre G2 phase. What will be the synonyms for G2 phase? Post S phase or pre M phase. What will be the synonyms for M phase? Post G2 phase or pre G1 phase. Question asked in any ways, you have to be in a position to answer, ma. Fine. You have understood the synonyms, right? Now coming to the M phase. Okay. Second one is what? M phase. In M phase, two things will happen in an orderly manner. Okay. What is that? First is karyokinesis. Second is what? Karyokinesis followed by cytokinesis. Fine. Karyokinesis followed by cytokinesis. So if you have to see karyokinesis both, it happens in a different way both in mitosis and meiosis. In mitosis though, it has four substages. What are them? Prophase, metaphase, anaphase, telophase. Whereas in meiosis, you have meiosis 1 and meiosis 2. Okay, you have meiosis 1 and meiosis 2, which is interrupted by an interkinase stage. Okay, interkinesis you will call it as, in between meiosis 1 and meiosis 2. What are the 4 substages in meiosis 1? Prophase 1, metaphase 1, anaphase 1, telophase 1. Okay. What are the 4 substages in meiosis 2? Prophase 2, metaphase 2, anaphase 2 and telophase 2. Very good. Okay. Cytokinesis, it takes place differently both in plant cell and animal cell. What type of cytokinesis takes place, we will discuss. In plant cell though it will be centrifugal, in animal cell it will be centripetal type of cell cytokinesis. Okay. So, what is the gist of cell cycle? It has 2 phases, interphase and M phase. Interphase has 3 subsequent phases. What will be pre-synthetic phase? G1 phase. What will be post-mitotic phase? Post-mitotic phase. G1 phase. Just look into the cell cycle and answer me. Okay. What will be pre-G2 phase? Pre-G2 phase. Pre-G2 phase is S phase. What will be post-synthetic phase? G2 phase. So, fine with all the substages ma. Now, we will look into what are the activities that are taking place actually. Okay. When it comes to cell division, okay, 3 types of cell divisions happen. In interphase, complete cell will be preparing for its division phase, right. Interphase, cell will be preparing completely for the division. Actual division phase is what ma? M phase. M phase is actual division phase. But is the M phase same for all the cells? No. For unicellular organisms like amoeba, some protozoans like paramecium, what will be the M phase? It will be just fission. Okay. That fission you will call it as the first one, it is called it as amitosis. You will call it as what ma? Amitosis. Okay. Amitosis means the cell will just divide into 2 without any spindle fiber formation. That is amitosis. You will usually see it in prokaryotes or unicellular organism. And second type you know it, that is mitosis type of cell division, and third type which takes place in germ cells, okay, that is meiosis. Let us get started with 3 subsequent phases of interphase. So, in cell cycle, if I have to divide the time period, a human cell will divide for 24 hours, of which 23 hours will be of interphase only, just 1 hour is of M phase. Okay. If you have to see yeast cells 90 minutes of cell cycle will be happening. Same way 90% of the time will be occupied with what? Interphase. It is as simple as it is to remember the statement beta, like for your NEET preparation. You are preparing years together for just how much time of exam? 3 hours 20 minutes time of exam, you are preparing years long. Same way a cell to divide just in 5 or 10% of total cell cycles time, it is preparing how much time? 90% of cell cycles time is occupied by what phase? Interphase. Okay. 90% of cell cycle time is occupied by interphase. Okay. Where the first phase is what? G1 phase. Quickly tell me the synonyms of G1 phase. It is called as post. If this is the cell cycle, you tell me this is G1, right? After G1 what phase? Nandu, you tell me Nana? S phase. Aisha, you tell me what is after S phase? G2. Followed by what? M phase. Now G1 phase is called as what? Post M phase, and it is pre. Remember. So in G1 phase what happens is most of the organelle duplication takes place. Most of the organelle duplication occurs. Okay, in G1 phase most of the organelle duplication occurs, and whichever RNA, whichever proteins, whichever DNA replication takes place in S phase. But whatever proteins, cellular constituents, RNA required for DNA replication will be synthesized in which phase? G1 phase. Because of most of the organelle duplication, what happens? Cell size will increase. cell size will increase. Got it, ma? So yes, G1 phase most of the organelle duplication occurs and cell size will increase. Next, tell me the synonyms of S phase. Post G1 phase and pre G2 phase. Post G1 and pre G2. What are the events that occur in synthesis phase? In synthesis phase, what happens is chromosomal duplication happens. For example, if you have to consider this as a cell, okay, this is a cell and inside that you assume this as a nucleus, fine. Inside nucleus, these were the chromosomes. How you will count the number of chromosomes? By the number of centromeres. At the center, centromeres are there, right? It is equal to number of chromosomes. How many chromatids are there here? 1 and 2. 2 chromatids, right? How 4? These are the arms of chromatids. This is one chromatid and these are 2 arms of the chromatid. As such, how many chromatids you have? 2 chromatids in the beginning of the S phase inside the nucleus. So, in S phase, what are the 2 major events that are happening in the nucleus is DNA duplication, okay. DNA will duplicate but chromosome number is same, okay. DNA duplication occurs but chromosome number is same. In cytoplasm, what happens? Centriole duplication occurs. What happens, ma? Centriole duplication occurs. This is the actual centriole which is in the cell. In S phase of the cytoplasm, one more centrioles are synthesized inside what? Centrosome. Don't get confused with the names of centrosome and centromere. Centromere is what? Primary constriction of the chromosome. Centrosome is what? Inside which centrioles are present. Got it? So, here what is happening in the S phase, ma? 2 things. One is DNA replication or duplication. Is there any increase in chromosome number? No. Chromosome number remains same. Second event is what? Centriole duplication. Second event is centriole duplication. Centriole duplication happens where, Nana? Inside the cytoplasm. Centriole duplication takes place inside the cytoplasm. Next, G2 phase. Here in G2 phase, you have to remember 2 major points, okay. Self-duplicating organelles or semi-autonomous organelles will replicate in G2 phase. Tell me what are the 2 self-replicating organelles? Yesterday we have discussed. Should I give you a hint? Double membrane bound organelles. Mitochondria and chloroplast. Very good. Mitochondria and chloroplast. Mitochondria and chloroplast. These are what, Nana? Self-duplicating or semi-autonomous organelles. Along with these 2, Golgi bodies also will duplicate. Golgi bodies will duplicate in G2 phase, okay. And whichever extra RNA protein synthesis is required for the M phase is also synthesized where? In G2 phase. Most importantly, tubulin protein synthesis happens in G2 phase. What is that? Tubulin protein synthesis. Fine, got it? So, what are the events of S phase? DNA replication inside the nucleus, centriole duplication inside the cytoplasm. Is there any increase in chromosome number? No, but DNA is doubling, right? And in G2 phase, what is happening? Semi-autonomous or self-duplicating organelles are doubling. Along with that, Golgi bodies are replicating. Next, tubulin protein synthesis occurs. Fine. And next, if you have to see, that is M phase. In M phase, first let us look into the mitosis. Here only I am telling you, mitosis is same as that of meiosis 2, or else we can say meiosis 2 is same as that of mitosis. Okay. And the well known statement, you all know that mitosis takes place in what? Somatic cells. Mitosis takes place in somatic cells. Let us look into all the 4 stages. How is it happening? What are the changes that are happening? First, in prophase 1. So, Just now I have told you that statement of NCERT that in S phase DNA replication is happening but chromosome number will not double. But the chromosome structure you will see at the end of prophase and metaphase. Till metaphase you will not see chromosome structure only, it is not visible, it is in the form of thread like structure. But to make you understand that only DNA is duplicating, not the chromosome number, he has used that hypothetical statement. Do not get confused with that. Okay? So, yes, here in prophase what happens is, the very first event is chromatin condensation. What is that, ma? Chromatin condensation. Fine. And the second most important thing is nuclear membrane, nucleolus, and all the organelles like endoplasmic reticulum, Golgi bodies will disappear at the end of prophase. Fine. And what happens? Third most important event in the prophase is the microtubules or spindle fibers whichever are present, these microtubules will radiate the asters to the opposite poles. What are these two, ma? Asters. Centriole duplication happens in which phase of interphase? Centriole duplication. S phase. Centriole duplication happens in S phase. Those centrioles will radiate, microtubules whichever are there in the cell, they will radiate the asters into two opposite poles. So, this asters moving to two opposite poles takes place where? In the prophase. So, these are the three events that you have to remember with respect to prophase 1. What is the difference between mitosis and meiosis? Yes, ma, I will tell you once the mitosis and meiosis concept is done, I will tabulate you the differences between mitosis and meiosis. Just listen to the class beta. Till then just listen to the class. After the completion of mitosis and meiosis, I will be telling you. So, in the metaphase if you see, all the chromosomes whichever are synthesized in the prophase, those chromosomes will come align at the center. Okay, all the chromosomes coming to the center equatorial plate, that phenomena is called as congregation. You will call it as what? Congregation. Fine. So, this chromosomes coming to the equatorial plate, how they are coming? These asters will radiate out the spindle fibers. Spindle fibers are connected to what structures of the chromosome? To the they are connected to the kinetochores. Okay. Kinetochores are two disc shaped structures at the sides of the centromere. So, to these kinetochores those spindle fibers are attached. Done. Now, third is anaphase. In anaphase what happens? Anaphase of mitosis, the centromere which is present here, the primary constriction, it gets splitted. Okay. Centromere splitting happens, and in anaphase the the chromatids of these two poles are there, right? They will move in different shapes. Okay. Those shapes are like V, J, V, L, J and I. Okay. Where metacentric, submetacentric, acrocentric and telocentric chromosomes. As such you have 4 shapes of the chromosomes. Here what you have to remember, you know, chromosome structure is clearly visible in which phase? Metaphase. Shapes of the chromosome is clearly visible in the anaphase. And at the end of prophase, you can observe that chromatid structures in the light microscope. Okay. When it comes, the question comes about visibility of chromosomes. At the end of prophase, what happens? The chromosomes will be visible under the light microscope. Clear structure of the chromosome you can see in the metaphase. Shapes of the chromosomes you will see in the anaphase. In telophase what happens? Half set of the chromosomes are here, half set of the chromosomes are here. These chromosomes will get decondensed again to what? Chromatin. They will decondense to chromatin. Fine. Nuclear membrane, nucleolus and endoplasmic reticulum, Golgi, everything will be reappeared at the end of telophase. So, these are the consequent events from prophase to telophase. Clear na? Fine ma? So now there is one point for you people to remember, that mitosis can happen both in the haploid and diploid cells of plants. Okay, in plant cells mitosis can happen both in the haploid and diploid cells. When it comes to animal cells, mitosis occurs only in the diploid cells. Okay, remember that point. Ma'am, is there any example? Okay, we need some example to remember, right? That example is connected to your chapter sexual reproduction in flowering plants, wherein from haploid megaspore that undergoes 3 sequential mitotic divisions and they form haploid embryo sac. Do you remember that? Haploid embryo sac is formed from haploid megaspore. So that happens, 3 mitotic divisions are happening in the megaspore. At the end in embryo sac, what you will see? 7 cells, 8 nucleated condition, where one egg cell will be there, 2 polar nuclei and 2 synergids, 3 antipodals, right? So this is formed from where? Haploid megaspore. Same in the case of microspore also, no? Haploid microspore will undergo 1 mitotic division, will form a vegetative cell and generative cell in the pollen grain. Fine. So mitosis can happen both in the haploid and diploid cells. Statement accepted, but only in the diploid cells of animal cells. Fine. Next, coming to cytokinesis. In animal cells, what happens is, what is the limiting boundary of animal cell? Cell membrane, right? So there will be a furrow that is formed at the center, which is bringing the cells close together like this. So it is from outside to inside. You are calling it as what? Centripetal. Centripetal type of division because of cell furrow. Fine. Whereas in a plant cell, as I told you yesterday, in between 2 cells, first what is formed? Middle lamellae. Okay, you can also call before middle lamellae, first initial wall that is secreted is phragmoplast. What is that, ma? Phragmoplast. This is synthesized from Golgi. Okay, and this gets matured to what? Middle lamella. Okay, this middle lamella is formed in between 2 cells. Next, close to middle lamella, what is formed next to middle lamella? Towards inside, primary cell wall is formed. Next to that, secondary cell wall is formed close to the cell membrane. So fine. From middle lamella, you tell me what will be there? Middle lamella, next to that primary cell wall, next to that secondary cell wall, and next to that what is there, beta? Cell membrane, and inside that nucleus. Okay. So this will be the order from outside to inside. Here what happens? The phragmoplast which is secreted here, it is moving to the opposite direction. So it is not from outside to inside, this is from inside to outside. So this you are calling it as what? Centrifugal. What do you call it as, beta? Centrifugal. Done? Fine. So have you understood both cytokinesis of plant cell and animal cell? Clear, right? Next, meiosis. In meiosis 1, meiosis 2, both are there. Once again let me recall you, you have what? Meiosis 1 followed by meiosis 2. Okay. In between meiosis 1 and meiosis 2, what do you have? Interkinesis stage. Okay. So meiosis 1 has prophase 1, metaphase 1, anaphase 1 and telophase, of which most important phase is what? Prophase 1. Okay. Most important phase is prophase 1. This prophase 1 will have 5 substages. Okay, it has 5 substages. What are they? Leptotene, zygotene, pachytene, diplotene, diakinesis. LZPDD. You can remember like that. Okay, leptotene, zygotene, pachytene, diplotene and diakinesis. Here what you have to remember, ma, the sequence, the events that are happening in each substage of prophase 1. First, if you have to see the leptotene, okay, in leptotene what happens? Same way chromatin condensation will happen, okay, chromatin gets condensed and next what happens? The asters will be moving to the opposite poles, fine, and here the chromatid structures will be aligned to the nuclear membrane. So that is like a bouquet stage, it looks like a bouquet or spireme stage. So that is why you will call liptotin as bouquet stage or spireme stage, fine. It is also called as bouquet stage or spireme stage, that is because the chromatins whichever are condensing, those thread like structures are attaching to nuclear membrane and they are looking like a bouquet, okay. Now what happens in zygote? Zygote is the most important phase beta. In zygote you have to remember few terms, I will tell you what is that. First, synaptonemal complex, bivalent formation, homologous chromosomes, all these terms are used where in the zygote. Firstly, you tell me what are homologous chromosomes. Do you have any idea about homologous chromosomes? So, I will tell you, see, meiosis happens in the germ cells, right. So, for example, there is a male gamete and a female gamete, haploid, haploid sperm cell, haploid egg cell. So, whatever traits our parents have, here comes the concept of genetics. Just listen to me for another 3 to 4 minutes, you will understand. Whatever traits that our parents have, we are getting that. Few of them are like variant traits of them, most of them like similar. Like how we are getting that? For example, this is the chromosome from father, let us suppose X of father and X of mother, okay. So, as such in the cell you have many chromosomes. What are the total number of chromosomes? 46 chromosomes you will be having, right. So, amongst that, those chromosomes which have one trait or one character will have how many variants? 2. One trait or one character, ma. For example, hair color is there. Father, let us suppose he is having black hair, mother let us suppose she is having grey hair. Like that, dominant and recessive will be there, no. Each trait is having 2 variants. As such, for example, let us assume tallness, okay. One is dominant like tall, one is short, okay. Those 2 chromosomes in which the alleles are at the same location, these are called as homologous chromosomes. So, in liptotin stage all chromatids are formed, right. Those 2 chromosomes which has same allele location for one trait, they will come together. Then this chromosome you are calling it as homologous chromosomes. Yes, Vishnu, you can ask the doubt, ma. I will clarify it. You just drop the message in chat box, I will be answering your doubt. So, the 2 chromosomes which has alleles at the same location or same loci, those are called as what? Homologous chromosomes. Those homologous chromosomes coming at one plane. So, here can you see these 2 are homologous chromosomes. As such, these are what? Sister chromatids of one chromosome. These 2 are what? Non-sister chromatids of homologous chromosomes. Fine. So, homologous chromosomes coming together will form what? Synaptonemal complex like this. If they come together only, no, in the next stage they can recombine. Okay. How recombination is happening? Exchange between the alleles. Okay. Those 2 alleles which belong to one trait, they come close together, these are called as homologous chromosomes. Homologous chromosomes have 2 sister chromatids on the same chromosome, non-sister chromatids on the other chromosomes, and the 2 homologous chromosomes coming together, you will call it as synaptonemal complex. Understood, right? Okay. And now 2 homologous chromosomes coming together, you will call it as a bivalent. One bivalent is having how many chromatids? 4 chromatids, right? 1, 2, 3 and 4. You have 4 chromatids for one bivalent. One bivalent is equal to 1 tetrad. 4 chromatids means 1 tetrad. One bivalent is equal to 1 tetrad. Understood? Is that everything clear from here? Okay. Everything is clear with respect to zygote. Vishnu, is everything clear with respect to zygote? Yes. Good that you know me. Now, Paketine stage. Okay. Now, homologous chromosomes got close together. Two non-sister chromatids of homologous chromosomes. Like, see, this and this are non-sister chromatids, but they have alleles at the same location for one trait. They will get recombined, forming what? Recombination nodule. Okay. In Paketine, what you will see, ma? Recombination nodule. Recombination nodule. Okay. And recombination is facilitated by enzyme. Recombination or the crossing over is an enzyme mediated process. What is that enzyme? Very good, Aisha. Recombinase. Okay. The enzyme is recombinase. This is an enzyme mediated process. Next, diplotin. In diplotin, what happens, you know? The chiasma is formed here. During the recombination, two chromosomes coming like this is called as what? X-shaped chromosomes. They form, crossing over gives rise to this shape. Fine. So, here in the diplotin, what happens is synaptonemal complex gets dissoluted. What happens? Dissolution of synaptonemal complex. Synaptonemal complex. Okay. After recombination, okay, recombination is done. Now, these two chromosomes moving apart will give you an X-shaped structure. That X-shaped structure is seen where? In the diplotin. So, yes, X-shaped structure is called as chiasma or chiasmata formation. So, in diplotin, what are the events you have to remember, beta? Chiasma or chiasmata formation or X-shaped formation and dissolution of synaptonemal complex. You will remember, right? Yes. So, one more point to say you here. In diplotin, okay, this is the stage where most of the oocytes will stop their cell division. For example, in the menstruation cycle, for each menstruation cycle, one egg will be released, one ovum will be released. So, all the ovums are prepared at the time of the embryo or fetus formation only during the female birth. But then the oocytes of most of the mammalian cells will stop their cell division at which stage? Diplotin stage. You have to remember that point. Okay. And next about diakinesis. In diakinesis, you have to remember the events like terminalization of chiasmata. Terminalization of chiasmata or chiasmata. Fine. The X-shaped chromosomes will be moving apart. And at the end of diakinesis, what happens? Nuclear membrane, nucleolus, Golgi, endoplasmic reticulum, everything will be disappear. So, whenever I am using the term at the end of prophase, nuclear membrane, nucleolus, endoplasmic reticulum, Golgi, they will get dissoluted. It does not mean that they are disappearing somewhere else. They are losing their structural integrity. So, at the end of prophase 1, the cytoplasm will get more dense and thick, because all the organelles are losing their what? Structural integrity for giving the space to cell division. Okay. In metaphase 1, what happens is, you can find two homologous chromosomes in two places, right? Here also all of them are coming together. But in metaphase 1, you can see bimetaphasic plate. Two metaphasic plates are formed at the center. What do you call it as? Bimetaphasic plate in the metaphase 1. And where are the spindle fibers attached to the chromosome? To which part? Kinetochore. Okay, kinetochore, which are disc shaped structures over the centromere. Fine. Now, anaphase. Okay. In anaphase, what happens? Anaphase 1, major point you have to remember is there is no centromere splitting. Okay, there is no centromere splitting. Fine. In anaphase 1, there is no centromere splitting. What happens is, the two chromosomes of the homologous chromosomes, one will be moving towards this pole and the other towards this pole without any centromere splitting. So, here you can see bivalent chromosomes are getting splitted without any centromere splitting. They are moving to the opposite poles. So, where do you see the bivalent chromosomes? In bimetaphasic plate in the metaphase 1. Bivalent chromosomes moving to the opposite poles in anaphase 1. In telophase 1 you can find the univalent chromosomes. Next. So, at the end of meiosis 1 you can find 2 daughter cells. Okay. Reduction of chromosome number happens in which phase? Here is the question. Reduction of chromosome number happens in which phase? In mitosis, is there any reduction of chromosome number? No. Chromosome number will be same, like how many chromosomes are there in the parent cell will be same in the daughter cell. Haploid to haploid, diploid to diploid. Where in meiosis we all know that major point that chromosome number gets half, chromosome number gets half or reduction of chromosome happens, right. So, reduction of chromosome number is happening where? In the anaphase 1, anaphase 1, because if you see in metaphase you have complete set of chromosomes in one cell, complete set of chromosomes. In anaphase 1, as they are moving to opposite poles, we consider this as one cell and this as other cell. Okay. So, chromosome number got half. You consider from this diagram only, how many chromosomes can you see? 1, 2, 3, 4, 5, 6. 6 complete set of chromosomes. Here if you have to see completely, one more I am drawing here. Three and 3, right? 3 chromosomes towards this pole and 3 chromosomes towards this pole. Then keep that statement clear in your mind. In meiosis, chromosome reduction happens and that is specific to which phase? Anaphase 1. Clear ma? Next. So, here in the meiosis 2. In meiosis 2, as I told you already, meiosis 2 is same as that of mitosis. Okay? Meiosis 2 is same as that of mitosis. So, now if you see, these are the clear differences between mitosis and meiosis. Vishnu, you can look into here beta. Okay? So, these are the 2 differences between sequence of the differences between mitosis and meiosis. Mitosis occurs in somatic cells, meiosis occurs in germ cells. Okay? And here nucleus divides only once. Here how many times karyokinesis is happening? 2 times. Karyokinesis 1, karyokinesis 2. Daughter cells, how many are formed at the end? 2 daughter cells in mitosis. How many daughter cells are formed at the end? 4 daughter cells. Daughter cells will be same as, the ploidy will be same as that of parents. If it is diploid, it will be diploid. Here they are haploid. And meiosis, in any cell it may be happening beta, plant cell or animal cell, parent cell is diploid. Okay? Meiosis happens only in the diploid cells. Next, it occurs more frequently, meiosis occurs less frequently. Daughter cells form from somatic organs. Here they are formed from, they form gametes. Here they form somatic organs. And here you just have 4 phases. Here you have 5 substages in prophase 1. Number of chromosomes will not change. Here reduction of chromosome happening in which phase? Very good. Anaphase 1. Here no crossing over. Here crossing over occurs. What is the other name for crossing over? Recombination. Recombination. That is what process? Enzyme mediated process. What is the enzyme? Recombinase, like that. Recall it multiple times so that you will remember. Okay. No crossing over in chromosomes. There is no crossing over. Next, here crossing over occurs. This is called as equational division because there is no reduction of chromosome. This is what? Reductional division because there is reduction in chromosome number. These are the differences of mitosis and meiosis. So, the entire chapter I have kept in front of you within just half an hour. Okay, it is almost like half an hour or 40 minutes. It is clear to you, no? Now we will solve few questions, few of them are NEET PYQs, few of them are statement based. Let us get started. First question, answer this ma'am. Online students also you can answer. Who is this student lucky? There are no inappropriate messages should be given in the comment section. Anything related to subject you type ma. Yeah, first question. How many chromosomes will the cell have at G1, S and M phase? G1, S and M phase. M phase here you can consider mitosis only because he did not mention specifically it is meiosis. Same type of question appeared in TGAP set. Yeah. Option A. No? Look into it properly Nandu. Draw a cell cycle. Okay. For example in G1 phase. Okay. Next S phase and next G2 phase and M phase. G1, S, G2 and M phase. Like that in G1 phase, let us say there are some number of chromosomes. The chromosome number will be same in S phase, same in, if it is mitosis it is same in, look into the options properly. Option B. Okay. If at all here he is specifically asking you meiosis, in G1 phase it is 14, in S phase it will be 14 at the end, but at the end of M phase or meiosis it will be 7, reduction in chromosome number happens. It is very simple, no? Try to apply the content which you know, try to apply the knowledge which you already have. This is how you have to do, okay. Second question, look into this. This occur in NEET 2025. I have given you the example also. What is the mature female gametophyte in angiosperm? Embryo sac. Just now I have taught you this. Try to answer, if not, no problem, I will say you. Online students, you can also answer beta. So, you see how many meiotic and mitotic divisions need to occur, okay. How many meiotic and mitotic divisions need to occur for development of mature female gametophyte from megaspore mother cell in angiosperm plant. So, I already told you the questions will be connected from cell cycle to sexual reproduction of flowering plants, right. So, here is the answer. I will tell you about chelacel and micropyle and everything when I am dealing that chapter. As of now you remember, from one megaspore mother cell ploidy will be diploid, right. One megaspore mother cell will undergo meiotic division, one meiotic division, or you can also call it as reductional division, forming what? 4 megaspores. It forms 4 megaspores. Of these 4 megaspores we know 3 will be degenerated and 3 will be degenerated, 1 will be functional. This will be haploid megaspores, yes or no? They will be haploid megaspores. This haploid megaspore will undergo 3 sequential mitotic divisions. How many ma? 3 sequential mitotic divisions, okay, forming the embryo sac, okay, forming 7 celled and 8 nucleated condition, where you have the egg cell and polar nuclei at the center, fine, and these are 2 synergid cells, antipodals, fine, got it. So, now you tell me how many meiotic and how many mitotic divisions? 1 meiosis and 3 mitosis, if he is asking you from megaspore mother cell. If in the question he is asking you from only megaspore, haploid megaspore, how many mitotic divisions occur to form the female gametophyte? 3 mitotic divisions, right? Are you here? It is 3 mitotic divisions from haploid megaspore. If the question is from megaspore mother cell, then 1 meiotic and 3 mitotic divisions. Fine, ma? So, here what will be the answer? Option 1. Clear to everyone, right? Online students also? Now, look into the statement based question. NEET 2024 appeared question this is. You have to answer. Statement 1, is it true or false? True. Correct, no? Good. Statement 2? Correct, right? In which stage terminalization of chiasmata will happen? Diakinesis. Good. So, here both the statements are correct. Good. I am happy at your response. Good. You are getting the point. Last question. Look into it, ma. Oh. This is connected to here, but I will tell you, no problem. This is terminalization of chiasmata. Here the appearance of chiasmata in which stage? You have to organize it in sequence. One. Okay. We will first locate the substages. Okay. Thin thread-like chromosomes with a beaded structure, where you will see in the liptotin, right? Liptotin. Second, appearance of recombination nodules in pachytene, right? Third, formation of bivalents or tetrads, zygote. Very good, Aisha. Next, terminalization of chiasmata, just now you people said diakinesis. Appearance of chiasmata, diplotin, that X-shaped structure in diplotin. Now you tell me the sequence. Tell me the sequence. First option 1, followed by 3, followed by 2, followed by 4, followed by 4, followed by 5. It is B. Once Nandu look into beta. 1 tarvata formation of recombination nodules in pachytene, no ma? 1, 3, 2, 4 and 5. Fourth option, okay. Option D. Fine. Got some grip on this chapter? Fine. Tomorrow we will look into one more new chapter and we will explore that. Fine. Yeah. Thank you.